So we've seen the process of expression regulation in bacteria, and bacteria obviously more simple organisms with smaller genomes, fewer genes, and really as a single cell navigating its environment, less diversity of range of options available to each individual cell. So getting into eukaryotes, I want us to start thinking about mostly multicellular eukaryotes. They're more complex organisms. They've got organelles inside of the cell, so even a single celled eukaryote is more complex than a bacterial cell because it's got organelles to worry about. And then also the possibility that these many cells of a multicellular organism have to really work with their own cellular environment. So let's see how that plays out a little bit here. One of the things, so as we think about eukaryotes, we've got to think about more complexity in time, but also in space. So if we go back to the fruit flies, Drosophila, this is an organism that's had a lot of study with respect to development. So going from an egg to a larva, going through larval stages, and genetically, it doesn't look like much, but what you can see on this image are these bands of coloring that's artificially added to, it's actually sort of molecular painting where they've identified regions where certain genes are active. So fruit fly genes get some interesting names: giant, NERPs, even skipped. What these are doing is they're laying down the pattern for building the whole fruit fly. So the embryo starts early on, it starts as this oval mass of cells. And as it goes through time, the cells become the beginnings of the different organs that are going to show up in the adult fruit fly. So one of the important division types that happens is these organisms become segmented. So they've got segments in their larva, and as they go on to develop into adults, those segments become important body segments with different functions. So on the right-hand side, you can see basically what has happened as a result of those divisions being laid down early in development is there are segments, these T thoracic segments and the A abdominal segments and the C the head segments, these different parts of insects going from start to finish, from head to tail. They become different parts of the fly. So the cells contained in this C3 mass of cells doesn't look like much here, but they get the instructions for becoming the proboscis, as it's shown there. Thorax and so on, these 1, 2, 3 thoracic segments, those are where the limbs are coming out from. So these insects that have 6 legs or 3 pairs of legs, each pair comes out of a different thoracic segment. So identifying as T1 gets it off to the start of making the front limbs. If you remember from telling your sexes apart in fruit flies, the forelimbs are the ones where the front limbs are where the males have their sex combs. So there are different programs for the cells that are part of those. And through cell division, through elongation, through metabolic changes, those different cells become, they take on the functions of the parts of the body that they need to have. So parts that are emitting pheromones or producing hairs or developing eyes. All these things are contained as instructions for the cells that belong to a certain segment. So hopefully you can kind of see how the complexity really increases. This is the same organism. This is all fruit fly. And the nuclei of the cells in all these segments should be the same. So the DNA is the same across this organism. But each of the cells is going to get different instructions for how to use its DNA depending on where it's located. So getting back to the idea of gene expression, where not every gene is required all the time, the abdominal segment here is going to have genes related to developing eye pigments. It's just not going to use them. And well, you get the point. So as I said, more complex in space and time for multicellular eukaryotes. In time, as they're developing into a multicellular organism, they might have times during development when certain genes are required. And then in space, as we've been discussing, different locations on the body where different genes are required. If you want to think within your own body, you've got organs, and your organs have different functions. You've got portions of your lung that have genes required for lung function. You've got parts of your pancreas with genes required for pancreas function. All those genes are contained within your genome. They're just not all used in all of your cells. So this is one of the big reasons we have bigger genomes in multicellular organisms, but also requires now a more complex expression regulation. So call back to this one. We saw this before in the ways that gene expression can be controlled. And we're still going to see a lot of preference for early control. So again, if the process can be controlled early on, that means that the cell can save some energy. So if it just never does transcription, then it can prevent wasting energy on an RNA transcript, wasting energy on translation for a molecule that it might not need at the end. Okay, so we have seen a lot of transcription regulation. We'll see a little bit more as part of this topic, but we did spend a lot of time on that in the bacteria. So this first one is showing us one of the expression regulation levels where the DNA can be wound up or loosely packaged. And so if the DNA is tightly packed, as it's shown on the top image here, this is tightly packed DNA. It can be packed so tightly that it's not accessible to the enzymes that do transcription. So if the DNA is there, but it's not accessible, then the genes contained on those pieces of DNA also won't be transcribed. So sometimes you can see this if you look at chromosomes. This is a chromosome squash from Drosophila, and it has these characteristic banding patterns. So early on, before we had the modern techniques for looking at DNA, this was a way that people would get familiar with pieces of chromosome that had their genes that they were interested in. So there's sort of a unique barcode kind of arrangement of regions on these chromosomes that are tightly packed or loosely packed. And tightly packed also means that they're going to show up darker because they have denser DNA that's going to hold more of the stain molecule, and it shows up darker. So it's a combination throughout these chromosomes of tightly packed and loosely packed, and I've highlighted a couple, a really dark region right next to a really light region that you can hardly tell is part of the same chromosome. The loosely packed regions are accessible to enzymes to do transcription. The tightly packed regions would not be accessible. They don't have to stay that way forever, but there are some regions that are more often found as packed up regions. So one of the ways that DNA can be packed up or relaxed is through chemical modifications of the DNA or of these histone molecules. So DNA, when it's wrapped up, is wrapped around histone proteins. That's what's shown in this green cylinder here. And the blue is the DNA. So the DNA is wrapped around those. And after it wraps around these, then it bundles up tightly together. You can see how this is very well organized and also pretty tightly packaged. And the cell can go between tightly packed DNA, which is shown on the left, and loosely packed DNA, which is shown on the right, through a couple of mechanisms. The first one I'll talk about here is called acetylation, and it's a modification to the histones themselves. So they call it histone acetylation to put them on, or deacetylation to remove them. So the cell can direct enzymes to add some acetyl groups or to remove some acetyl groups, whether it needs to activate or inactivate certain gene regions. So that's probably the level of detail I want to go into there. Another way that control can happen is through adding methyl groups. This is a modification to the DNA itself, and turns out that methylation is a modification to the cytosine type of nucleotide. So up in the top right here, we've got cytosine and methylated cytosine. This is a methyl group, CH3. And by adding that, that's part of the signal. When DNA is methylated, that's part of the signal to pack it up tightly. So a little bit opposite of what we saw with the acetyl groups. The acetyl groups cause DNA to be more relaxed, and the methyl groups cause DNA to be more packaged up. We got methylation and demethylation. Again, don't worry too much about the detailed names of these. They're getting a lot of attention in research these days, but I think it's still more names than you need to worry about. So I mentioned methylating the cytosine, the C DNA nucleotides, and it turns out that methylated locations are often found at this C next to a G. So in the same reading direction as it shows here, 5 prime, C then G. That's a location that could be methylated. And regions that are methylated often exist in what we call these CPG islands, or regions that are dense with the pattern of C and G. So this region here shows CG, then CG, and those both have methyl groups. And nice thing about CG is it is complementary to CG. So if we're reading the same direction, 5 prime to 3 prime, the bottom strand also shows CG, and then CG, and this would be a CG here as well. And the ME here is methyl groups. So methylation, acetylation can silence up DNA. Just a cute little cartoon I wanted to throw on here. Another gene silenced. All right, so this is, I took both of those images and modified them from a paper that talks about how methylation and acetylation work together, as I've mentioned before. So really whatever it takes to achieve the end, it could be methylation, it could be acetylation, it could be a combination of both to cause transcription to be enabled or prevented by packing up the DNA. I think I'll probably just link this video in the, I'll put it on Canvas next to the other video. This is a video showing the process of X inactivation. So way back when we talked about X inactivation as part of the dosage compensation for females that have 2 X chromosomes to get the same amount of gene expression on their X chromosome as males have with 1 X chromosome. So this shows how that inactivation is based on packaging up the X chromosome very tightly, and how it's important to be able to do that for the whole chromosome. There we go. Another topic related to inactivation by methylation is, again, kind of a big topic these days called epigenetics. And epigenetics is a term that refers to differences among individuals that are not due to their DNA sequence, but there's still differences in gene expression. Their epi is sort of on top of, so it's almost genetic. If we stick to genetics, like require that word to mean DNA sequence, which we often do, then these are differences that are not due to DNA differences in the DNA sequence, but they can be due to differences in the DNA expression or the DNA accessibility. There's a lot of attention being paid to this. So we have techniques for observing what pieces of DNA are methylated. Comparing those between individuals that have been in different situations is a common research approach. As with this study, this is a study where they took people and they subjected some to exercise and left some to rest. And they saw that there was a significant difference in the expression, I should say specifically the methylation of a couple of these genes that are related to muscle function. So what I think is neat about this is that we do see evidence that a person's genetics are not entirely in control of what they become. So I think sometimes you learn about genetics and you think, oh, I'm bound to be, I'm already pre-programmed to be a certain way, and there's no way out of that. And it turns out that there's actually, it's possible to change what your body can do with its DNA, depending on how you lead your life. So exercise is one of these big things that can be hard for people to get started with, and they're not sure if it's going to pay out in results. But you actually could long-term kind of affect how your DNA is accessed by your cells based on what you do in your life. So we see positive examples, exercise leading to what people usually think of as positive effects. You see negative effects, Some certain habits like smoking, other habits can have negative effects or undesirable effects of methylation across the genome. You're in control a little bit more than you might have thought at the beginning of this class. Turns out also that epigenetic changes can be inherited. So you inherit the DNA from your parents. Turns out you also can inherit the methylation states of the DNA from your parents. So this is a study where they took mice, and you can see the mouse on the top left here is a male mouse, and the male mouse is not contributing much to the egg except the DNA from his sperm cell. And what it shows is that these mice are being trained in the parent generation. They have an experience where they smell a certain odor molecule, and that's associated with a mild shock. So they're getting trained to basically avoid that odor as a signal that shock is going to come. And so they're averse to the odor. And this mouse has a child with a mouse that hasn't experienced that. And the child of that mouse, having not been exposed to any shocks in its lifetime, is predisposed to be a little more startled in the presence of that odor molecule. So here we're showing, instead of a shock, this is a loud noise. So they test the startle response. How likely is this mouse to be frightened by a loud noise in the presence or in the absence of this odor molecule? And so it significantly, it shows that it's trained to be a little more worried about that odor smell, even though it hasn't had any bad experiences with that, and would not have had any training from his father. So this mouse is just completely new to the experience, but genetically has this epigenetic change in its DNA that causes it to be a little more worried about that chemical. So I guess one thing I should point out here is that we don't really talk about it in this class, but a lot of behaviors, so neurological changes in organisms, those can be genetic. So we don't often think about them with humans, but there are innate behaviors. So animals can be born with a lot of good strategies for navigating their environment, even if they're not trained on those. And those can be basically imprinted genetically. So either genetically or epigenetically, you can maybe see the adaptive benefit of this. So mouse learning that something is dangerous in its environment through an association that's basically molecular association. Okay, so down to one generation, this mouse is more startled in the presence of the odor, and still another generation. So this mouse down here is 2 generations removed from the mouse that was ever shocked in the presence of this odor molecule, still a little more commonly, a little more often startled in the presence of the odor molecule. So this can be something that's passed on continually, the epigenetic state of DNA there. This is one getting back to humans. We talked about studying twins in order to learn more about genetic differences. This is a case that doesn't arise too often. They had a chance to send an astronaut into space who had a twin. So they sent one astronaut into space and they left the twin on Earth. So Scott Kelly is the name of the astronaut who went into space. And they tested his DNA against his brother. So again, as in with the twin studies, up to that point of going into space, they would have led a very similar life to each other, even though they're grown men. They had a childhood of very similar experiences. And certainly they've both lived on Earth for all of their life, as I hope most of us can say. And they found some differences. And they found differences in gene expression. So this is related to methylation and epigenetics. And I also like to point out, related to this news item, that there were a lot of news reports that didn't get it right the first time around. So there were initially a whole bunch of stories that came out and said, this guy went to space and his DNA is 7% different than it was before he went into space. Well, hold on, not quite true. So the DNA is the same. The nucleotide sequences are the same, but the methylation states, the expression states are 7% different. So still a pretty substantial difference, but possibly one that could be reversed. Methylation states, as we've seen, can go on and then off again and certainly something they want to study as to what changes, what changes in your genetic makeup, your epigenetic makeup, as you go into space. I haven't heard anything of like a important gene that was discovered as a result of this. All right, so that was a big function here, just affecting whether the DNA can be accessed. Similar to what we saw with the bacteria, now we're going to talk about the transcription, really control over the process of transcription itself. And we've seen these before, and actually I ended up cheating, and this is also an example from bacteria, but not quite the one-to-one relationships we saw before. If you have a set of genes that all need to be turned on in response to, say, the same environmental condition, there can be an advantage to activating a single transcription factor that turns on all those different genes. Maybe I can draw what I'm talking about here. So if you have a variety of genes, and let's say they're all needed as you, I don't know, go into winter. So genes, the gene expression helps you to regulate your body temperature when it's cold outside. So if you turn on or enable transcribe a transcription factor, then it would be able to turn on transcription of all these genes that are needed at the same time. So lest I give you the idea it's turning everything on, I'm going to limit us to 3 of these genes and say that this transcription factor can turn on expression for A, C, and D. And if this is going to be adaptive, then A, C, and D should have a common function or a related function responding to similar kind of experience, environment. And if they all get turned on in the presence of this one transcription factor, that's one way to make a change across the genome in certain kind of cells or under certain kinds of experiences. So this example, this is called the FER protein, which stands for ferric uptake regulator. Ferric is another word for iron ions. And just a set of, there's some other candidates, but I picked some of the ones with strong evidence, genes that are required for basically regulating iron uptake. So as a bacterial cell finds itself in an environment with iron, it can turn on this transcription factor. So basically transcribe and translate the transcription factor, and then that in turn turns on these other genes with the functions you see here. So just kind of a flavor for what people are discovering with this kind of thing. It opens a big door. So you often find tens of genes that are upregulated in a certain environment, and then it's up to you to find out why they're all upregulated, what kind of function they have related to that. Still a lot of gene discovery underway. Okay, so this helps. So like in time or space, if we turn on the expression of different genes in time, this can be respond to conditions or maybe a certain time in development. Or these could turn on at a certain different location in space. So time and space. An example might be genes that are all turned on in a certain kind of organ, or a combination of time and space. So let's say you've eaten a meal and you got some food coming through your digestive system. Your body is responding to the situation in time where there's now food present, and then also in space where there would be genes in your digestive organs that are turned on in response to that. So there's a big diversity of transcription factors, a whole lot of research going into those. Basically, the transcription factors connect the signals of what's going on outside the cell with how the cell should respond to that in terms of translating new enzymes. It's a very dynamic process and certainly hard to encapsulate here. But another part of that is also kind of shown here with the complexity of the promoter. And then farther upstream is a region we call the enhancer region. We've seen this before as these different regulatory proteins get involved. There's lots of action around the promoter, which is shown in this goldenrod color. But there also can be some regulators that attach much farther upstream in these enhancer regions, different colors associated with different molecules. And so it doesn't even have to be, as we saw here, it doesn't even have to be one transcription factor, but maybe a combination of transcription factors. Again, just without going into details, thinking about the possible complexity of what all could be turned on under these scenarios. So these are the signals that are possible to interact with. This is the whole transcription complex interacting with the promoter and also portions of the enhancer to enable transcription. So in general, in eukaryotes, this is more complex than we saw in bacteria, but also probably more complex than we can make a whole lot of sense of. But whatever if you're interested in this and you get into the field of molecular biology, you certainly can dive down into whatever process you want to study and learn more about it. For example, this one we saw a long time ago, also the SRY transcription factor. That was the sex determining region on the Y chromosome. And if you remember, we saw that SRY, the simple presence of that SRY gene causes a person to develop as a male. And the absence of the SRY causes a person to develop as a female. And maybe at that time you thought, well, that's a big effect for one gene, and it certainly is. But it happens because it's helpful to have that on and off switch, presence or absence of SRY. And SRY in turn regulates a bunch of other transcription. So it's turning on transcription of some genes. It's also turning off transcription of some genes. So this diagram, I like this one a lot, it just boils it down very simply to ovarian genes, testicular genes. So basically, genes for becoming male are turned on by SRY. Genes for becoming female are turned off if a person has SRY. SRY also turns on transcription of SOX9, which then turns on other processes, still consistent with that same result of on for testicular genes and off for ovarian genes. And all of this, again, if you don't have the SRY, this whole thing doesn't happen, which means that it's kind of the default is ovarian genes are on, testicular genes are off. And so you got to switch between male and female. And then just another complex image here showing how there are some extracellular processes or some signaling events that can result in transcription. So don't worry about any of the details, but we've got basically the outside of the cell and the inside of the cell, and all these abbreviations are proteins that are interacting with each other, and ultimately some of them interacting with the DNA to cause transcription of certain genes. So cells are responding to their environment, and a lot of times they respond, I think I can say genetically, so they respond by turning on transcription, enabling enzymes, doing their central dogma to make those enzymes exist where they didn't before. All right, moving down our diagram. So it's possible to have control at all these steps, but we're going to focus on a few of these. This is one for degrading RNA. There's been a lot of new research in this direction. We're finding a lot of pieces of RNA that are important. I'd say previously it was harder to zero in on small pieces of RNA, but now they can be observed better using our techniques. And so there are natural processes happening within cells and also useful artificial processes. And this is one, what's shown here is a process called RNA interference, where we can artificially sort of manipulate a process that happens naturally. So by adding double-stranded RNA, this is something, it sets off a cascade, basically kind of sets off an alarm inside the cell. Because double-stranded RNA is not something that it typically has, but double-stranded RNA is sometimes encountered as a result of a virus infection. So if you have viral RNA, sometimes it makes double-stranded RNA as part of its replication. And that's like a giant red flag for the cell. Like, I've got to break this apart or I'm going to have a viral infection. So the cell responds to that by destroying all pieces of RNA that look like the double-stranded RNA. It cuts it up, and then it also uses a piece of that to train itself to find other pieces, is what's showing in this diagram. So this happens if you've got viral double-stranded RNA, and you can do it artificially by adding artificial double-stranded RNA of the gene you want to turn off. So some piece, even a small piece of the mRNA, the coding region of the gene you want to turn off, if you add double-stranded RNA of that gene, the cell is going to destroy that. And that's become a useful research tool for intentionally turning off genes really on demand, which is kind of cool. All right.